Hello everyone, in the previous video I discussed about the key components of cell-mediated immunity. I will provide the link of the first part of cell-mediated immunity in the description box, or you can just watch it in my playlist. Now let's understand the process of how antigens are presented by MHC molecule. For a T cell to recognize a foreign protein, and this is basically we called antigen, the antigen must be, let's say this is antigen, and this antigen is recognized by T cell. So for a T cell to recognize an antigen, the antigen must be broken down into small pieces or small peptide fragments. That this is basically small. This is basically antigen, and this antigen is broken down by small fragments, and then these fragments bind to either class one, bind to either class 1 MSC molecule or class 2 MSC molecule. This whole process is basically called antigen processing and presentation. And whether an antigen associated with MSC class 1 molecule or MSC class 2 molecule depends on how the antigen enters the cell. So the antigens basically enters the cell by two types of origin. One is external origin, external origin, and the other is internal origin. So this is basically internal origin. So those antigen which enters the uh, enter by external origin is called exogenous antigen. That is exogenous antigen, and those antigen which basically produce in the cell is basically called endogenous antigen. So now let's see the exogenous antigen. This exogenous antigen is basically come from outside of the cell and enters by let's say enters by endocytosis or phagocytosis. And this exogenous antigen basically produce end of the day this exogenous antigen basically bind to the MHC class 2 molecule, and MHC class 2 molecule basically present this antigen, and this happens in basically antigen presenting cell. So and we know that antigen presenting cell, there are many types of antigen presenting cell like macrophages, dendritic cell or B cell. Now let's see the process of exogenous antigen formation. So this is basically a antigen, or let's say a bacteria or a virus particle. This let's say this antigen is ingested by this antigen presenting cell, and then antigen presenting cell form an endosome, and then in the cell there will be lysosome. The lysosome fuse with the endosome and form endolysosome. This endolysosome then break down this antigen into small peptide fragments. These small peptide fragments then attach to the this MHC class 1 molecule. Then this MHC class 1 molecule went through the surface of the cell, and now here this cell surface contain MHC class 2 molecule. This MHC class 2 molecule is basically presenting this antigen, and now let me change the color. Okay. And now this antigen is basically recognized by T helper cell, because in the previous video we know that this helper T cell is basically recognize the antigen by MHC class 2 molecule. It is and it is basically restricted to all types of T helper cells. So this is basically the process of exogenous antigen, or how this antigen is presented by T helper cell. And now in the term of endogenous antigen, let's see the process. Let's say this is a virus, and this virus inserted its DNA into this cell, and here endogenous antigen is basically produced from all types of nucleated cell. This is not basically produced from antigen presenting cell. This antigen presenting cell is always uh, uh, produce exogenous antigen, and all types of nucleated cell produce endogenous antigen. So now let's see the process. This virus inserted its DNA into the cell, into the nucleated cell. Then this DNA will this DNA will go through the transcription process and make its RNA. This RNA then go through the translation process, and this RNA will produce protein. Now this protein are degraded into small peptides, and this degradation happens in the endoplasmic reticulum. So remember this here, this viral protein degraded in the endoplasmic reticulum, where they bind to class 1 MSC molecule. So this degradation happens in the endoplasmic reticulum, and then these small peptides bind with the MHC class I molecule in the endoplasmic reticulum. Then this is transported, this is go through the Golgi complex. This Golgi complex will form a vesicle, and this vesicle then go to the surface of the cell. And now here in this surface, the MHC class I molecule present the peptide or viral protein to the T site, or we say that cytotoxic T cell. So this is also restricted. So this thing also restricted that cytotoxic T cell always recognize antigen from the class 1 MHC molecule. Or cytotoxic T cell is also called CD8 plus T cell. This CD8 plus T cell recognize the antigen class 1 MHC complex, or this whole thing we can call this is a complex. This is a peptide class 1 MHC complex. We can say that. So in that way, the T helper cell or T cytotoxic cell recognize the foreign particle or recognize the foreign antigen. So in the cell-mediated immunity, when T cells are activated, they proliferate into two main groups. One is effector T cell and other is memory T cell. Now see that. Let's say T cells get activated. And how these T cells get activated? These T cells are get activated by the MHC class 1 molecule or MHC class 2 molecule which present the antigen, or it's get activated when it recognizes the foreign antigen. And after the T cell gets activated, it differentiates into mainly two groups, two types of T cells. One is effector T cell, and the other is memory T cell. And this effector T cell are the cell that actively participate in the immune response. This include helper T cell, T cells, or this is also called CD4 plus T cells, because there is CD4 plus surface protein on the helper T cell. And the other is cytotoxic T cell. This cytotoxic T cell also called CD8 plus T cell, because this cytotoxic T cell contain CD8 plus surface protein on their cell surface. In the next video, I will discuss how helper T cell and cytotoxic T cell get activated or basically act in the cell-mediated immunity.